So you're very welcome, everyone, again to the Green Era Hub webinar series. My name is Barry Crosby, and I'm one of the communication officers for the Green Era Hub project. If you've been here with us before, you'll know that the Green Era Hub project officially started in September 2022 as a coordination and support action of the European Commission under the Horizon Europe programme, and it currently represents 15 EU ERA nets, co-funds and self-sustained initiatives in the field of agri-food and biotechnology. We started these breakfast clubs here last year in October, and we've had 4 very successful series so far. The first one was Circularity in Agriculture. The second series was related to biotechnology to advance sustainable solutions for bioeconomy. The third was Sustainable Food Systems, and the one that we've just finished was Precision Agriculture Technologies for a Sustainable Future. If you're interested in listening to the talks related to any of the topics from these series just mentioned, our recordings are now available to view on our YouTube channel. Now, for the next 3 months, we're focusing on the topic of plant health and protection, where we'll be hosting a series of expert-led discussions aimed at fostering a deeper understanding of plant pathology, integrated pest management, and sustainable agricultural practices. And just a few housekeeping announcements. Please do feel free to type your questions in the Q&A chat section, which I've enabled for everyone. I'm going to do my best to address as many questions as possible within the time that we have today, which is 1 hour. If at the end of the session you have a question for either of the speakers, please do remember to address the speaker in the question. Now, today we have 2 panelists joining us, Dr. Panagiotis Melonas and Dr. Evelyn Van Hesche, and we'll open our session this morning with Dr. Melonas, currently head of the Scientific Directorate of Entomology and Agricultural Zoology at the Benaki Phytopathological Institute in Athens, where he also serves as alternate Director General. Dr. Melonas' main expertise lies in insect behavioral ecology, integrated pest management, and biological control. With over 1,450 peer-reviewed publications and more than 900 citations, Dr. Melonas is a prolific researcher, author, and field expert whose work plays a crucial role in developing effective integrated pest management strategies that leverage biological control to sustainably reduce pest populations of plants. He's an active member of the Plant Health Panel of the European Food Safety Authority and serves on the advisory committee for PhD candidates, as well as reviewing for research programs and scientific journals. Dr. Melonas' talk is entitled Fruit Volatiles for Fruit Fly Infestation Detection, which is part of the Fresco Phytosanitary Research Initiative. Dr. Melonas, thank you very much for accepting our invitation for our webinar series. I'm going to stop sharing my screen now so you can start sharing your presentation. Thank you very much.
Thanks again for inviting me in this series of presentations about plant health. As you already said, my name is Panagiotis Milonas. I'm an entomologist in the Benaki Institute in Athens, Greece. And over the last few years, I've been working on fruit volatiles and how we can these volatiles be used for detection of an infestation, particularly on fruits by fruit fly species. So I'm trying to explain why the volatile organic compounds, why you are interested in these compounds in the volatiles, and how can we use these volatiles for detecting an infested commodity or fruits, either at import but also during export of fruits to other markets from the European Union. And I'm trying to explain the defense mechanism of the plant, because this is where the volatiles are related to. Connection of fruit flies and herbivore-induced plant volatiles. And I will present some of our results, and finally the electronic nose concept and our results on this subject. So to start with, is plants have to deal with a lot of herbivores that are attacking them in the environment, and they have two ways to do that. Either with a direct plant defense, which means that they directly have an impact on the herbivore itself, which means that using toxic metabolites or other physiological things that they have a direct impact on the herbivore, or indirectly plant defense, which means that they mainly enhance the effectiveness of the natural enemies with various ways, like providing food for them or shelter, or through the herbivore-induced plant volatiles. And by herbivore-induced plant volatiles, we mean volatiles that would be produced as a response to attack by the plant, attack by a herbivore. And these volatile organic compounds can either be produced in advance or produced only after the attack of the herbivore. In both cases, the result is that the profile of the volatiles emitted by the plant differs when it is under attack by herbivore. And why that is that? Now, think a complex environment where the plant exists, and there are many, many things that they interact with. There are other plants in around. There is competitor plants or parasitic plants. There is attack on below ground, other herbivores above ground. So there's quite a complex environment in the open field. And the natural enemies, the carnivores, parasitoids, or predators, need to find their prey. And their prey is usually something small in a very large scale environment. So the prey itself emits its own volatiles, but it's a small quantity in a big environment with a lot of other stimulants around. So they need something else, something to be also reliable, but in a large quantity. So the plants in an evolutionary context have evolved to respond to attack to attract the natural enemies to locate the herbivore, and so the plant would benefit from that. So our approach is to exploit this behavior that the plant emits volatiles under attack when they are under attack by the herbivore, in a way to detect the volatiles, the herbivore as a natural enemy would have done in the nature. Now, this herbivore-induced plant volatiles, there are different organic compounds. It can be mainly terpenoids or other fatty acid derivatives, phenylpropanoids, benzenoids, and others. And they can be emitted either at the site of the damage where the herbivore itself is feeding, or can be systemically produced from other plants or other parts of the plant as well. And you also know that beside the herbivore-induced plant volatiles, you now have the case of oviposition-induced plant volatiles, which means that a plant can respond even quite early of an infestation, of an attack, when it is even by the oviposition of an insect on the plant. So as I said, over the last few years, we've been working with fruit flies, which are a very important pest worldwide, notorious pests that attack a great variety of fruits around the world. We mainly work with Ceratizis capitata, the Mediterranean fruit fly, which occurs in European Union, but is a Canadian species for many parts of the world and important markets for the European Union, like the United States or China. And also two Bactrocera species, Bactrocera zonata, the pitch fruit fly, and Bactrocera dorsalis, which is the oriented fruit fly. And you can see in the map, especially for Bactrocera dorsalis, that is expanding its distribution in the world. And there are also findings or incursions in Europe the last few years. But it's still these two species of Bactrocera zonata and Bactrocera dorsalis are important quarantine pest species for the European Union. There is already a lot of publications that deal with the volatile organic compounds being emitted by different fruits when attacked by different fruit fly species. So we know already that there is a response of a fruit being infested by different volatile species, but there was so far so intense study of the emitted organic compounds by the specific fruit fly species that we targeted. So why do we care particularly for the volatile organic compounds or the herbivore-induced plant volatiles? So we want to detect this during inspection. And although we can monitor adults with traps which are baited and with lures in the traps, these are mainly targeting the adults. But when there is an import, then phytosanitary inspectors, they need to check the fruits. And most of the cases you have the fruits will be infested by young larvae or even eggs, as what we can see in these pictures are eggs, which are laid just beneath the skin of the fruit. And in fact, the inspector will need to do a destructive sampling in a large proportion of a commodity, and needs also to be quite experienced to locate and identify this infestation by fruit fly. So we want to speed up this process by taking advantage of these herbivore-induced volatiles, and we focus on these three species, and with an overall goal to have a cost-effective, rapid, reliable method for inspection particularly during import controls or even for export quantities. So what you need to do as a first step is to collect these volatiles that are emitted by the fruits, isolate and identify them with analytical chemistry methods, and maybe at some time to also explore if there are affecting the behavior of the insects, if they have a real impact on the behavior of the targeted insects. Now I will go through a few of our results, so the different combinations of fruit fly species and fruits. And what you can see here is for oranges, for example, it's a chromatogram. The upper one is when you have an infested one, and the bottom one is when it's not infested. And what you see is that compounds that have been detected in both cases, there are quite large differences in the amounts that are emitted. So in this case, we have in the infested part, in the infested fruit, lower amounts of this group of volatiles. So as I said before, overall, the overall profile of the infested fruit is different from a non-infested fruit. And again, for the oranges, what we have seen is that particular compounds can be used to characterize these differences. And we see, for example, the limonene is increasing the first day just immediately after the infestation, but then decreases overall to similar compounds with a non-infested one, where other compounds like the oxymene is high from the beginning till also through the development of the larvae in the fruits. And all this in combination with other compounds, we have this what we call a fingerprint of an infested fruit versus a non-infested one. And this we can use it with a chemometric analysis to find out, to group the different groups of infested or non-infested fruits by the specific fruit fly. This is also important. We have a fruit specific response and a fruit fly species also specific response by the fruit. I continue now with the other fruit that we worked, peaches, and again we detected a lot of compounds. Again, compounds that appear in both cases, infested and non-infested. Then there are differences in the amounts emitted by the two different groups, infested and non-infested. In this case, again, we have some compounds that increase over the time of the keeping the fruits in the lab. and others remain at the same level. And altogether, again, we were managed to group them in infested and non-infested. In some cases, more clear, and in others, not so clear. But that depends a lot of also the fruit species, particularly peaches, because they have this maturation process also during the storage time of the experiment. And finally, we work also with apples. And again, we've seen these differences among the infested and non-infested fruits based on specific compounds. And here, for example, we can see the difference for a particular compound, farnesene, which is on an infested fruit by Ceratitis capitata decreases, but versus the control. But when it is infested by Bactoceras ursalis, increases versus the non-infested one. So the first conclusion from all this that the fruit fly infestation do trigger emission of herbivore-induced plant volatiles. That's the first thing that came up with these studies. And our analysis of the data showed that it was able to discriminate between healthy and infested fruits based on these herbivore-induced plant volatiles. And for each fruit, a particular group of compounds, or sometimes even one compound, can be used to characterize different stages of the infestation, the beginning or later parts of the infestation. But that's okay when you are working in the lab. The thing is that you need to take that in a practical situation of an inspection. And there you need something more portable, like an electronic nose, which is constructed in a way to mimic the mammalian olfactory system. And it's based on kind of sensors to identify different gases or vapors. They have the sensors, and these sensors, they come into contact with a specific compound, and then the chemical properties or the physical properties change, and you get this response of the sensors, which is a typical fingerprint. So it did use a commercial available one, electronic nose, to train it with a different group of infestations on versus non-infested fruits by the fruit flies. We constructed then the fingerprints, and then we analyzed the data to see if this is a reliable method for detection of a fruit fly infestation in the field. Just to give you an example, that electronic nose are used at the moment for various ways in the food industry, particularly like to detect shelf life on eggs or elasticity of the food or maturity of strawberries and various other uses, particularly in the feed industry. So I'm going to go through a couple of slides to show you our results on fruit fly infestation and electronic nose. And very quickly, you can see that, for example, on peaches and Bactocera zonata infestation, 20 degrees storage time, we had some really good results. Please pay attention on this yellow part, 85% correct identification. Then for the med fly, again with peaches at 25 different temperatures, close to 80. Again, close to 80 with peaches. Apples, we tested also at different temperature conditions. Even better description, 94, 80. Just go through that very quickly, and don't to stay a lot. But the overall is that with electronic nose and using the right algorithm, because you need to explore the data set that you have with the correct algorithm, which in our case is the key nearest neighborhood, we came up with an identification close to 80% of the infestation. So in a way, the electronic nose offers a rapid analysis. It's cost-effective and non-destructive, but at the same time, it has some disadvantages at the moment, like it needs a lot of calibration and maintenance and a lot of initial investment to start with. But there's a lot of studies and efforts in Europe, particularly to develop specific sensors for plant volatiles to target and detect infested fruits. And so just to conclude that we know now that post-harvest fruits do respond to fruit fly infestation, and it's possible to discriminate in healthy versus infested ones based on this herbivore-induced plant volatiles. And electronic nose is a promising technology for detection of these fruit flies on commodities. Yeah, thank you so much. I mean, that was that for me.
Yes, thank you so much, Dr. Malonis, for such an interesting and insightful presentation. I just want to remind everyone that the chat box and Q&A section is open, so please do feel free to type a question for Dr. Malonis at any point over the next few minutes. Dr. Malonis, just while we're waiting for questions from our audience, I just had a couple of questions from your presentation that I was just keen to ask, and I was just kind of wondering about. I know you touched on this a little bit during the presentation, but I was wondering about the kind of environmental factors and the role that environmental factors play in the emission of fruit volatiles. And I wonder, could you maybe just expand on that just a little bit for us, please?
Well, you're right. Environmental conditions they will, do play a role in the emission of the volatiles, and that was one of the reasons why we tested particularly the electronic nose at different temperatures to see if it works, how it works, or how it will behave under a different temperature scenario. So you need to keep in mind that we're talking about trade of fluids, and most of the time they will uh, be stored in relatively low temperature because they want to maintain it for during the transport. So that's why we tested also at low temperatures. But yes, higher temperatures will also affect the higher emission rate. But at the beginning, what they were trying to do is to prove that over the development of the insect inside the fruit, because you need to keep in mind that the larvae after infestation, they continue developing inside the fruit. So we wanted to prove that at the different developmental stages of the insect inside the fruit, we can still see these differences coming out from the volatile profile of the plant. This is also a limitation for the electronic nodes because they might be affected by the environmental conditions like humidity or temperature. And although the e-nodes can detect, but there might be some masking effect of these factors in the actual conditions. So we need to do a lot of testing on that in real-time conditions.
I see. Fantastic, thank you. The other question I had related to cross-species interactions, and I was wondering what role do fruit volatiles play in attracting beneficial organisms, such as maybe predators or parasitoids of fruit flies?
Well, interesting, but parasitoids, they have this, there are many species of parasitoids, and a lot of them, they have the capacity with their long ovipositor to locate the larvae which are underneath the skin of the fruit, and their long ovipositor inserted into the fruit on the larvae, which is under the under the skin of the fruit, so they can parasitize their host. So they can use these volatiles to locate where the host is. It's a response because the plant is, in theory, it's called the cry for help theory of the plant. The plants want to have their enemies, the natural enemies, to stop the infestation.
I see. Excellent. Thank you very much, Dr. Malonis. Just one other question. In terms of economic sustainability, I was just wondering about how or if fruit volatile detection systems improve the economic sustainability of fruit farming?
The detection of an infestation is very important for any fruit fly management. And the starting point here is we want to avoid the introduction of Canadian pests, for example, in Europe, in the European Union. And these are priority pests for the European Union because they will have a major economic impact if they are being introduced into European agriculture. So to maintain our farmers' profit and European agriculture being resilient when we have an attack from invasive species, it's good to stop that before they come into EU countries. And one way to do that is through the inspections and input controls. And at the time, now this takes a lot of time and effort by the inspectors. We want to make their job easier and more profitable for all by identifying all commodities that are coming into the EU.
Very good, very good. Thank you. Just one question just coming in from our audience, and it reads, in a storage place, there is a broad variety of different fruits or vegetables. How far will this disturb the detection of an infestation of a specific fruit fly in a specific commodity to be checked by inspectors during import inspections?
Well, think about like how the inspection is done. The inspector will have to isolate the specific commodity to be inspected. Like when you have import of I don't know, mango, then the inspector will check the commodity that's coming through the borders. And the protocol now is to take a large sample and do the structured sampling on its fruit of the sample to see if there are any infestations. So what we're trying to do, not only us, but also other groups in the EU, is to have a technology to help this inspection. And the technology will isolate the particular sample that you are looking at. And then this is somehow isolated from the other environment. And the machine, through artificial learning and these algorithms, will be able to identify the specific compounds that characterize the infestation of the particular fruit. At the moment, almost all fruit flies for Europe are under quarantine status.
Excellent, Dr. Malonis. Thank you very much. Just another question coming in from our audience. Can fruit volatile analysis be used for early detection before visible infestation?
Yeah, exactly. That's exactly what we're trying to do, because you have seen also a response at a very early stage of infestation, even the same day or one day after the oviposition by the adults. So yes, it's possible to discriminate between an infested and non-infested fruit at a very early stage of infestation when visual damage is not obvious. Yeah.
Excellent. Thank you very much. And just a last question just before we finish. Can this method distinguish between different fruit fly species, example from the same genus?
Well, that's obviously more difficult, but our results did show that there is a different response of the fruits based on the different fruit fly species. Just to remind, we were targeting Bactocera dorsalis and Bactocera zonata, two species of the same genus. And in several cases, we have seen that the response of the fruit was different depending on which fruit fly species was infesting the fruit.
Dr. Malonis, thank you very much once again. Unfortunately, we have to leave it at that because it's just gone past 9:30. But thank you so much for your excellent presentation and for the questions from our audience. I think it was a really excellent and very insightful presentation. Okay, so joining us now is Dr. Evelyn Heesche, who is currently Scientific Officer of Nematology at the Netherlands Food and Consumer Product Safety Authority, where she specializes in policy advising, research, project management, and diagnostics within the nematological and phytosanitary sectors. Dr. Van Heesche's work has contributed significantly to the development of national policies focusing on enhancing plant health and safety in agriculture. Her expertise and understanding of nematode biology and ecology in particular has informed the development of sustainable pest management strategies, while her advancements in diagnostics have helped improve pest identification accuracy, enabling effective responses to phytosanitary challenges. Today, Dr. Van Heesche's talk is entitled Meloidogyne Enterolobii, and I hope I pronounced that correctly, Survival Under Temperate Climate Conditions and Distribution Within Europe, which is also part of the UFresco Phytosanitary Research Initiative. Dr. Van Heesche, thank you very much once again for accepting our invitation to our webinar series, and we're absolutely delighted to have you today. So without further ado, the floor is yours. I'm going to stop sharing my screen now, so you can start sharing yours. Thank you very much.
Okay, sorry, my voice. Thank you very much for this nice introduction. And I will try to share my screen now. First, yeah, as I said, many thanks for the introduction. And it's for giving this opportunity to tell a bit more about the research we perform on the survival of Meloidogyne Enterolobii under the colder temperate conditions as we have in the Netherlands and in Belgium. I work at the Netherlands Institute for Vectors, Invasive Plants and Plant Health, and that's part of the Dutch NPPO. And probably one of you of the audience have seen already my talk of Marleen, my colleague Marleen Botermans. She told a bit about high throughput screening and viruses. So I'll give no introduction anymore about our institute. I will go directly to what will be in this presentation. As I'm not sure if everybody is quite familiar with Meloidogyne Enterolobii, I will give a short introduction first. And I focus why it's a quarantine nematode in Europe, and also some background of associated import requirements. And then I will tell you more about the research goals and activities we are performing in this UFresco project, and some first results we have, as it is a 3-year project. At the right, you will see a bucket with potato plants with 10-liter jars of buckets, and we use this in our outside trials. Idea of how a trial is looking like. So Meloidogyne Enterolobii, it's a big risk for Europe due to the breakthrough of resistance it can do in tomatoes and bell peppers. So besides that, it has a very broad host range. Important crops like potato, cucumber, tomato, peppers, but also soybean and tobacco are hosts. And besides, also ornamentals as ficus and petunia are hosts. And yeah, we don't know yet much also about hosts in temperate climates. A lot of the research is done in the more warmer climates. But what we do know of the work of Brito, and one of the American scientists who's working a lot of Meloidogyne Enterolobii, is that a garlic and peanut and grapefruit are non-hosts. So there are a few exceptions, luckily. So it can complete his life cycle in only 6 weeks at 20 degrees. So when it's a bit warmer, it goes even faster. So Brito also did some research when the root nuts clearly are appearing for inspections, to see for inspectors if they can't see root nuts or not. That's around 10 weeks at 22 degrees. So that it will take some time for inspectors to see the roots of their galling or not. Unfortunately, also big yield reductions are there around 65% in tomato and 50% in cucumber. So that's quite a lot of damage. And one of the disadvantages, especially in greenhouses, is that there's a possible spread with their irrigation systems. There is evidence already for Meloidogyne incognita and Hapla. And also we did a small trial with Meloidogyne Enterolobii, and we see also that it can be spread. So that's also a big problem. Like you see on the right side in the picture, that are bell peppers, which are really, they're in the irrigation system standing. And there is a lot of problems there. You see them really hanging. And the last thing about Meloidogyne Enterolobii is that also diagnosis is quite difficult. Often tropical root knot nematodes are in mixed infestations, so different species in the same soil or a root system. So it's very difficult to identify, especially also morphological. Evolutionary, they're quite close to each other. So there's only small differences. You have to be really an expert to identify them. So here is the life cycle of Meloidogyne Enterolobii, or Meloidogyne in general. If you look at the effective juvenile to G2 stage, it goes into the roots, and it becomes, when it's becoming a female, a feeding cell is induced, and due to that inducement, there is a cell division starting, and that is creating those galls. The size of those galls or nuts is really variable. It can be very big to very small, depending on the species and crop combination. The tropical species in general have some bigger galls than the temperate ones. For instance, at the right side, you see above a tomato root with also Meloidogyne Enterolobii on it. You see under some big galls, there are already several life cycles of females and juveniles there, but also from the small ones. Also potato, you see just under the peel, you see the brown dots. That's the necrotic tissue in which a female is laying. From 2022 April 11th, it has become the EU quarantine status. That's because to prevent the European agriculture to major economic damage it can cause, and also to be able to try to have at least a free trade of a trade of free of Meloidogyne Enterolobii to prevent further spread. To safeguard this, we have specific import requirements. So plants into the European Union need to be coming from a free country or a free area, or they has to be grown their entire life in soil actually not in soil, or soil with organic parts, or the growing medium has to be entirely free of peat and cacao, and fumigated or heat treated. And besides those three things, it has to be in physical isolation from the open soil. Another option is that it has to be coming from a free production site, and then first, before export, it has to be visually inspected, the roots. As you see on the right, that's the EPO distribution map. Meloidogyne Enterolobii is already quite spread over the world, especially in Southern and Middle America, in Africa, and also in Asia and Australia, some countries. So yeah, why are we doing this research, this Fresco project? The last few years, since it is a European quarantine nematode, we have some findings in the Netherlands and Belgium at import and export inspections. And as it is our task as an NPPO, we have the responsibility that entering and leaving plant material coming from the European Union we have to safeguard our agriculture So we want to be sure that we're not spreading further. But unfortunately, most research is done at tropical temperatures, so we don't know anything about survival in the temperate climate as the northern part of Europe. Besides that, we are dealing with a climate change. The winters are becoming more warm, so we have less frost, especially in the Netherlands and Belgium and that region in Europe. The winters are less colder, becoming temperatures below zero or less and less there. So we really need to have more knowledge about the probability and of the establishment and survival in the open field to prevent this possible spread. So what are the activities we're doing within this Fresco project? First thing is we're doing a lot of research on the epidemiology of Meloidogyne Enterolobii. So we're going to perform life cycle and survival experiments at various temperatures between 5 and 20 degrees, 20 or to 25 degrees in climate rooms as well as outdoors, and also in the absence and presence of host plants. And after those experiments, we also assess the infectivity of the survivals, the juveniles who survived this. Another thing is that we're going to monitor and map the prevalence of Meloidogyne Enterolobii in the countries of our partners. We're doing that by surveillance. The partners are Flanders Research Institute for Agriculture, the Austrian Agency for Health and Food Safety, and also the Murcian Institute of Agriculture and Food Research Development in Spain. The project is for 3 years and it ends in the end of 2026. So we already have some first results. These are the first results from ILVO from some survival experiments they did under controlled conditions. They checked the survival of infective juveniles, the G2s and egg masses, at 5, 12, 20, and 25 degrees. And afterwards, they assessed the infectivity of the survivors. You see beneath some pictures, some small tubes were put at different temperatures in the controlled conditions. And they were all inoculated with around 500 juveniles. So here you see the survival in the soil of the juveniles. What you see is that at 5 degrees, at 2, 4, and 6 weeks after starting, you still see around 30 to 40% survival. And this is without host plants, actually. So that's quite a lot. Luckily, after from 8 weeks on, you see that it's really decreasing. And after 10 weeks, you see that there is around 5% survival at all 4 different temperatures. And if we then look at the survivors from the different for 4 different temperatures, when we look at the right, we'll check afterwards the infectivity. And what you see then is that of those who have survived, only 2 to 3% are really infective. We checked this by putting a tomato on those small bits of soil. And after 1 week, we checked the roots of the meloidogynes there or not by staining and counting the meloidogynes in there. But yeah, 2 to 3% for a quarantine nematode where there is 0 tolerance, especially for 5 degrees, you see that there is less variation between the temperatures. It's actually still too much. So also this was performed outdoors in bigger tubes. You see in the picture on the left. They were stored outdoors, but not in the rain, so that there was not too much rain coming into no rain coming into the tubes, but further there are in outdoor conditions. And more or less the same experiment was performed, but then with a little bit more soil, of course. So there are here the results. And actually what you see is that in the first 4 weeks, the survival was quite high, till 80%. You see in the red line the main temperature at that time. That is between around 8 till 12 degrees. So yeah, 4 weeks of black fallow. Actually, what this will be in practice, there is almost all nematodes survive, but then you'll see a decrease. And from 16 weeks on, but that's already almost 4 months. So that's in practice, black fallow, no agriculture company is doing 4 months black fallow. But yeah, then you see a real decrease of the survival. But what you see at 28 weeks, there is no survival. But when we put some tomatoes on those soil, we saw at ILVO, they saw still some infective juveniles coming out. So this at 28 weeks is more a problem of the detection limit of our extraction methods, because there were still some survivors. So those were some experiments from ILVO. Now we also in the Netherlands are doing some experiments on survival also. Outdoors, in the first winter, we did a pilot with only potato. And now the coming 2 winters, we are doing the big trials with perennial crop. At the end, it was Buddleia chosen and potato. And we're starting these trials every year in May. Then we'll plant the potato or plant the Buddleia. And then at the end of October, the potatoes are harvested, and the crops of the tubers are the peels of the tubers are extracted and checked for Meloidogyne, and the soil, which is still there in the big plastic 10-liter buckets, is stored in 4 different conditions. So we'll just in the open field, so that's the real winter conditions in the Netherlands, in a climate room at 4 degrees Celsius. 4 degrees Celsius is actually the average winter temperature in the Netherlands, so that's why we have chosen 4 degrees. Another few replicates are stored in the same 4 degrees, but then in the second part of January, 2 weeks at 0 degrees, so a mild winter is simulated there. And the last one is also at 4 degrees, but then for 2 weeks at -8. So a real severe winter is simulated by then. And we sample, of course, but also the Buddleia at the end in October, but also then in spring to see after those 4 conditions, the survivors. So to know a bit which plants we could use and which varieties, we've checked as a pre-trial in the beginning some different potato varieties and some different rose varieties and Buddleia varieties. What we saw is that potato is quite a good host, although compared to tomatoes, of course, there's still a big difference. It's only 10–50%, but yeah, it's a good host. So we've chosen Infestations as it has a quite a little higher population density than the Seresta. But what we saw on rose, actually, that's a highly viable host. You see quite an influence on the variety you're using. And also, for instance, Balmoral, the one above, you see in yellow that there are jars which had no Meloidogyne at all, but there were jars which had even more than 7,000 in there. So it's very hard, variable, not, so we didn't select rose as a crop or plant for further experiments, and it wasn't a reliable one. Buddleia was chosen, although it's a minor host, but still we saw, okay, there is some maintenance. So, and as we don't have another perennial plant who is surviving the Dutch winters yet, we know as a proper host, this was our best option. We did this pre-trial with 2 different populations, one from China, one from Puerto Rico, but we didn't saw a difference between the populations. But this is the results from the pilot year on the potato. Now, the potato cultivation was very successful, as you see on the right. Very nice potatoes with lots of nuts on it. But what we do saw also was that the multiplication of Enterolobii was less than for Shitwoodi, but that's also in a way expected, as Shitwoodi is more used to the colder temperatures in the Netherlands. So that's from the first part of the work package. The second part is on the surveys. In the Netherlands, we started already in 2021 with surveying for Meloidogyne Enterolobii, first year on perennial grasses. We sampled 30 fields, different perennial grasses, no Enterolobii find locally, and also in the second year on the ornamentals as Hosta and Liriope, we also find no Enterolobii. It was performed that the plants were checked above ground on reduced growth, and if we saw some reduced growth, we checked the root system if we saw galls or symptoms, and then we collected the samples from the roots as well as from the soil, but mainly from the roots, and we extracted and identified them. The year later, 2023, we had our first infestation in the Netherlands. So that was on ficus. So we checked all 33 ficus companies in the Netherlands, and also in the same way, checking the roots. And also in those companies, we checked risk based on 19 other ornamentals. Unfortunately, 3 out of the 33 companies had plants positive for Meloidogyne Enterolobii. In total, we had, of those 33, in those 33 companies, we sampled 18 lots, 11 ficus, 5 callistemon, 1 Rhodomagera, and 1 Zelkova. Only 1 Zelkova and 3 ficuses were positive. When we traced forward and backwards those lots, also 2 companies were checked further, and also in those companies we found 2 Zachariaceae lots, 2 Cesiums, and 1 Zelkova lot positive. Actually, till that moment, Zachariaceae wasn't in the picture for us, as we didn't know that that was a host plant. So you see that looking further also gives more ideas of host plants. So yeah, this is quite some information. I want to thank Wim Wezema and Johnny Visser for the work they perform also within this project, and of course you as audience for listening to me.
Dr. Van Hesche, thank you so much for that excellent presentation. Really clear, really insightful, and super interesting. I just want to remind everyone that the chat box and Q&A section is open, so please do feel free to type a question for Dr. Van Hesche at any time over the next couple of minutes before the end of the webinar. Dr. Van Hesche, just before we go to questions from the audience, just a couple of things that I was very curious about just listening to your presentation. I was particularly keen when you were talking at the beginning about, you kind of touched on a little bit the differences, and there are very small differences in biological terms between Meloidogyne Enterolobii and other root knot nematodes. And I wonder, could you just expand on that a little bit for us, if you can?
Yes.
The key kind of biological characteristics, I suppose, that differentiate Meloidogyne Enterolobii from other root knots.
Yeah.
Thank you.
Now for Meloidogyne, especially the tropical ones, we're talking then about Meloidogyne javanica, incognita, and Meloidogyne oranaria, for instance. They're very closely related. And then what general characteristics are, where we look at when we morphologically identify, then we look at the length of the juveniles, especially when I'm now talking first about the juveniles, the G2 stage, as it is the infective stage. The whole length of the juvenile, the stylet length, the shape of the stylet knobs. So that's really looking, is it 2 micrometers or 4 micrometers in length? So you really have to look at it in 1,000 times magnification in that case. So that's you really need a good microscope.
Yeah.
That's essential in my morphology of nematodes, but also the tail shape, the length of the tail, the position of some organs like the hemizoonite. Is it laying above or beneath? the excretory pore. Those are characteristics where we're looking at them. But yeah, you need to have a lot of experience.
Yeah, absolutely. Thank you very much. In relation, the other question I had was in relation to the crops that are most affected by Meloidogyne Enterolobii, and I wonder, could you maybe just expand a little bit on that? Now, I know the focus from your presentation, the focus on studies has been kind of mostly in the southern hemisphere, and it's only kind of really we're starting to look at their impact in the northern hemisphere of recent times. But just from your experience and from your initial research, what crops are most affected, do you think, by Meloidogyne Enterolobii, and how do infestations impact on their yield and quality?
I think, yeah, that I know for in tomato and peppers, it's a really big problem. But I think also outdoors, yeah, it's called, and the common name is guava root knot nematode. So in guava, it's also a very big it can be a very big problem. Also, I don't know numbers about damage, but I think you're in the same range as I was talking about on cucumber and tomato. 50 to 60% of damage of lower yields are quite common, I'm afraid. So it's due to the big galling, you see that there is a lack of taking up of nutrients and water. So the plant is actually dying. So yeah, that's it's a really big problem.
Yes, absolutely.
And yeah, oh yeah, I don't know. Yeah, and soybean, for instance, it's also a very big problem. It can be.
Yes, fascinating stuff. And Dr. Venetia, I was just wondering about the kind of distribution patterns, and particularly, I was wondering about what methodologies can be employed, I suppose, to kind of map the distribution of Meloidogyne Enterolobii across Europe.
Yeah, that's not really my. I know I have colleagues who do that for me.
Putting you on the spot here.
Yeah.
That question. Apologies.
No problem. No, it's starting with performing the surveys, knowing where it's if it's there or not in your country, and then in combination with some surviving experiments, knowing how it can develop at certain temperatures. We know, for instance, that Meloidogyne incognita, that's a sister species of Meloidogyne enterolobii, is surviving at least 10 degrees Celsius. So you can make then distribution maps. I know colleagues from Slovenia, for instance, did it in a new fresco project called Melotrop. Yes, Melotrop it was. I'm doing too much new fresco project with Meloidogyne, so I had to think first. No, and I think that's also on the Ufresco page available, and you see there that my colleague Sasha, I think, Sister Circa, did a very nice job in combination with our French colleagues with making some distribution maps, and also with a prediction already with a 2 degrees Celsius raising of the temperature within Europe, how far it can be spread then for those, not for Enterolobii, we hadn't done that project yet, but yeah. I know for there is within we are also involved in that the European Horizon project, NemEmerge. The coming 3-4 years, we're going to do also distribution mapping of Meloidogyne Enterolobii. So hopefully, in 3-4 years, in combination with these results, you know a bit more about the chances that it can survive also in the northern part of Europe.
Excellent. Thank you very much. Just a couple of questions coming in from our audience. The first is, can nematodes possibly survive better at greater soil depths, or has this been investigated?
Nice question, Stefan. Good to see you here. The depth of the nematodes, yeah, it's variable. We know, for instance, for Meloidogynes in general, that they're not going that deep into the soil. For Xiphinema, it's going another nematode virus vector. It's going much deeper. So yes, but I don't know by head of for Meloidogyne. It's there has been an investigation yet in all those different layers. I think till 30 to 50 centimeters, it can be there. But as far as I know by head, Enterolobii is not going, of Meloidogyne in general, is not going further deeper. So yeah.
Excellent.
Probably somebody has investigated it, but yeah, it's not all in my head, unfortunately, and not always published. That's also a pity. Some research is done, but not published.
Yeah, fantastic. Well, it's an area that certainly needs more investigation and needs more research on. So it's fantastic that you're kind of leading the path in many ways. Just another question, just very quickly, just coming in. Which species of Meloidogyne are often intercepted in the Netherlands and in which crops?
Which crops? What we, of course, we're not checking everything. If I see for Meloidogyne, for Enterolobii, for instance, in the presentation I had quickly, I haven't read them all, but there are those 19 crops where we focus on the moment, of perennial ones then in this case. So you see that Ficus, Sagaretia, Syncytium, Zelkova, especially from regions like China, you have to be very careful and check them properly. So that's and crops. Yeah, of course, we have our seed potato industry is very large in the Netherlands. So there are no interceptions, but there are screenings before it's going to be exported. There we find, of course, some Chaetwoodii, Meloidogyne Chaetwoodii and Fallax. In some cases, Meloidogyne minor also. For if we look, let's see, in where have we done some surveys the last few years? Yeah, I have to look for that further. But that's what the quick answer is.
Yes, no problem. Thank you so much. And just very finally, Dr. Venetia, just one last question. A member of audience is asking, what control methods are available for this pest?
Control methods for Enterolobii, it's not tested yet, but in the Netherlands, for Chaetwoodii and Fallax, we do use inundation as control method. I think in the warmer climates, solarization can also be used if it's done, but that's also for inundation if it's done on a proper way, as in the literature is mentioned. And crop rotation using and for Meloidogyne, yeah, that are the things which can be used at the moment in Europe because there are no chemicals anymore allowed, or almost no chemicals. So yeah, that's only a very small amount of things we can do. So that's why it's so important to be very quick on the detection of Meloidogyne in your field. Because yeah, when you have it there, as you saw, this life cycle is only 6 weeks, and one female is producing 200 to 400 eggs, so it can go very quickly. But yeah, one of the things we do know also less about is about the survival. We're checking now with juveniles, but also we're going to try to check with the whole egg mass in the gelatin matrix to see if that also can survive maybe a bit longer because of the gelatin around it, because there is also less in literature known about it at the moment.
That's great. It's fascinating stuff. Dr. Venetia, thank you so much. I think unfortunately we have to leave it there. It's just 10:05. But thank you very much, everybody, for your questions. I think we could go on for another 15, 20 minutes easily.
Yeah, that's always the case with interesting
Absolutely.
topics. The same
Um,
as from Mr. Milanios.
yes, absolutely. I'd just like to thank once again Dr. Melonas and Dr. Venetia for giving us such wonderful and insightful presentations and providing us, I think, with a really great overview of their respective projects this morning. So thank you both very much for joining us. Our next webinar will be on February the 20th at the slightly later time of 3 p.m. Central European Time, when we will continue our new webinar series on plant health and protection. I'll be sending out the invitation to all of you in the coming days. In that breakfast club, we will have Dr. Rujing Wang from the Canadian Food Inspection Agency discussing the development of a seed identification guide and database for invasive plants and weedy species, and Dr. Khalid Djula from the Mediterranean Agronomic Institute of Bari talking about the detection and identification of severe strains of citrus tristeza virus. So again, thank you very much to our panels, and thank you everyone for attending and for such really fantastic questions. We hope you enjoyed the webinar, and we'll see you all again next time. Thank you very much.